Okay.
Good to go?
Yeah.
Lovely. So at the end of the Krebs cycle, we have 2 molecules of FADH2 and 10 molecules of NADH for every molecule of glucose inside the matrix. On the inner membrane, we have proteins called electron transport chain proteins. And these proteins will pump hydrogen from the matrix to the intermembrane space. But these proteins can only do this when they get energy from the electrons that we find from FADH2 and NADH. So let's start with NADH. This molecule is going to donate an electron to the first protein in the electron transport chain. And this pump will have energy to pump hydrogen from the matrix to the intermembrane space. The electron will move to the next pump. This pump will have energy, and it will pump hydrogen from the matrix to the intermembrane space. This electron will move to the next pump. This pump will have energy. It will pump hydrogen from the matrix to the intermembrane space. The next electron is donated. It will join to the first pump. This pump has energy. It will pump hydrogen from the matrix to the intermembrane space. And now NAD can go back to the very first stage in glycolysis and get recycled and used again. The electron moves to the next pump, and it will then pump hydrogen again from the matrix to the intermembrane space. This electron will bind to the first pump. Hydrogen will then get pumped through this protein from the matrix to the intermembrane space. This electron binds to this pump. This pump has energy. It will pump hydrogen from the matrix to the intermembrane space. And again, FADH2, or FAD now, can get recycled and used in Krebs cycle again. This electron will bind to the next protein, and that one will pump hydrogen from the matrix to the intermembrane space. This electron will bind to the next pump. This pump has energy, and it will pump hydrogen from the matrix to the intermembrane space. And this is going to continue. So eventually you find that we have what's called a concentration gradient forming. So we have a high concentration of hydrogen in the intermembrane space and a low concentration of hydrogen in the matrix, because the electron transport chain has pumped all the hydrogen from the matrix into the intermembrane space. Now the hydrogen protons do not want there to be this concentration gradient. It wants there to be an equal amount of hydrogen in the intermembrane space and an equal amount of hydrogen in the matrix. But the hydrogen protons cannot travel back into the matrix through the electron transport chain proteins. These proteins are one way only. They will only pump hydrogen from the matrix to the intermembrane space. The only way that these hydrogen protons can go back into the matrix is through a very special protein called ATP synthase, drawn here in red. ATP synthase will join together ADP and phosphate to make ATP. And you can find the ADP and the phosphate throughout the mitochondria. Every single time a hydrogen proton travels through ATP synthase to get back into the matrix, it gives ATP synthase the energy to combine ADP and phosphate together to make ATP, energy for the cell. This hydrogen proton will travel from the intermembrane space into the matrix, and it will give ATP synthase, that's the name of the protein, the energy to combine ADP and phosphate together to make ATP. Now this hydrogen will travel back into the matrix through ATP synthase, and this molecule will have the energy to join ADP and Pi together to make ATP. And this will continue happening until for every molecule of glucose, 34 molecules of ATP are made. Now what's really important here is that you realize that these electron transport chain proteins are now full. We need a molecule to come along and take these electrons from these proteins, because otherwise they won't work. And the molecule that does this is oxygen. Oxygen is really important for aerobic respiration, because it will take 2 electrons, 2 hydrogens, and combine to form water. Oxygen will come along, take 2 electrons, 2 hydrogens, and form water. Oxygen will come along, take 2 electrons, 2 hydrogens, and combine to form water, H2O. Now these electron transport pumps are free to bind to more electrons and continue pumping hydrogen from the matrix into the intermembrane space, therefore making a concentration gradient so that ATP synthase can work. Done.